Good day to one and all. Welcome to FDP programs conducted by Botany for you. Myself, Dr. Ganga. My topic of discussion is hypersensitivity. So, before going to the details of hypersensitivity, let us have a look on what are the immune responses uh, present in a host immune system. So, immune responses are effectively to eliminate the any sort of antigens or foreign body which is enter in our body. So this there are two major different types of antigens or foreign body which can considered as foreign in our body. So one is an endogenous and other one is an exogenous type. So what is an exogenous? What is the exogenous challenges which is faced by immune system? Are microorganisms, transplant cells, toxins, allergens, chemicals, extra which is comes external from external to our body. Whereas there are few endogenous challenges which our immune system faces are host tumor cells and aged host cells. That is the antigens or external foreign substances which is considered in our body, which is present in our body, which the immune system consider as foreign. And there are some other immune responses which help in. There are few immune responses which help in to abate this foreign pathogens or foreign bodies in our body. That is two major type of immune mechanisms, like innate immunity and adaptive immunity. So innate immunity is further classified into two types: cellular immunity and humoral immunity. Cellular means that is cell mediated and humoral means antibody mediated. So innate cellular immunities are basically done by natural killer cells, macrophages and neutrophils. Whereas humoral immunity is done by by means of complements, lysozyme, fibronectin, hormones, beta lysins, and other polypeptides. Also, against virus, we have interferons. Against tumor, we have tumor necrosis factors and bacterial since against bacteria. And we have also have adaptive immunity, that is also have cellular type and humoral type. Cellular, as we all know, it is T cell mediated, and antibody humoral means B cell or antibody mediated. And there are other mechanisms called allergy, because for our own self antigen, it should not be dictated or degraded. For that, to remove the self detection or degradation of self, we have some tolerance mechanism we call as allergy. The two types of cell tolerance mechanism, that is central tolerance and peripheral tolerance. So today, in addition to that, immune response at times in an inadequate manner or misdirected manner. So this is called the immune dysfunctions. So malfunction of immune system, that is not it's not good for our body, or it is something deleterious to our own cells. So those mechanisms are due to some infectious disease, hypersensitivity, graft rejection, or graft versus host diseases, autoimmune disorders, and immunodeficiency. So today we are looking on such type of immune dysfunction reaction, that is called hypersensitivity. As name indicate, it is hyper means something extra, extra sensitivity. So that is immune responses are extravagantly produce some sensitivity reactions. So moving on to the introduction. Hypersensitivity is an immune response mobilizes an array of effector molecules that act to remove antigen by various mechanisms. This is that is remove. It has to our cells, our body is trying to, immune system is trying to eliminate an antigen by producing an array of molecules. Somehow it has to be getting eliminated. So a lot of immunological responses or reaction comes together to eliminate the foreign pathogen. that could be in turn deleterious to our own host tissues or damage our own cells. So that is hypersensitivity reaction. So that is hyperactive immune response is hypersensitive. Under certain circumstances, the is some inflammatory response which have due to this immune mechanism, it produces some inflammatory responses that result in significant tissue damage or tissue injury, some diseases, and or ultimately sometimes can lead even to death. So it is as I said before, it's an exaggerated or inappropriate immune mechanism which is harmful to host itself. That is hypersensitivity reaction. So first this was observed by Monaco. The Prince Monaco first observed the deleterious effect on jellyfish on birthers. So subsequently later the Portrayer and Richard suggested that a toxin responsible for this effect, and they coined the name anaphylaxis. So generally hypersensitivities have two major components. First, it is a priming dose. So that's the first initial dose of antigen is essential to evoke an hypersensitivity reaction, followed by a second dose, that is shocking dose of the same antigen, result in injurious consequences. So for a hypersensitive reaction to evoke, it requires two dose of reaction, that is an initial priming dose and a shocking dose, that is a second dose, which the second dose could be deleterious reaction. So if you look into what sort of how it does it occur, that is in the case of innate immunity, how an anaphylaxis reaction or a hypersensitivity reaction occurs when a microbes comes in contact with the epithelial cells. So immediately they'll get phagocytosis, and it will activate some complements and natural killer cells. And this reaction is taking place within hours, and it will get degraded by the all these mechanisms together, and it's taken within hours. That is the innate immunity mechanism. When it comes to the adaptive immunity, these all recognize the same antigen, the same microbe, and there will be an antibody production against this microbe. In the case of T cells also, with the help of antigen presenting cells and some other few mechanisms, that will take few days, that is up to 7 days, and the effector T cells will kill these antigens. So this is a two type of mechanisms, innate immunity and adaptive immunity works. So if you look into the adaptive immunity, generally it is B cells and T cells carry out major functions, whereas in the case of innate immunity, the cells, that is immune cells, is taking place major function like phagocytosis, macrophages, natural killer cells, complement, etc. So these are the two major mechanisms how an antigen get eliminated in our body. So the hypersensitivity refers to the high undesirable reaction produced by normal system. So this hypersensitivity can also be called as allergic reactions or anaphylaxis reactions. So there are different terminologies comes in the hypersensitivity reactions. So it's a clinical condition or a type of disease that involve more than one type of reaction. So hypersensitive reaction can be a many type of conditions which array of reactions comes together. It could be either swelling, it could be either itching, it should be production of temperature. So an array of reaction cannot be, we cannot specifically say this is only hypersensitivity leads to only itching. So it can be any different kind of reactions, like say for example, itching is one type of hypersensitive reaction, and the swelling of body is a hypersensitive reactions, and the watering of eyes, nose, etc. is another type of infection. Sometimes it be a foodborne, like we get diarrhea, loose motion, etc. That is, and vomiting. That is also an hypersensitivity reactions. Sometimes it can fatal and leads to death. So different clinical manifestation will be there for an hypersensitive reactions. So now how it has to be classified, hypersensitivity reaction has to be classified into 4 different types. Type 1, type 2, type 3, type 4. That is, among this type 1 up to 3 is antibody mediated and type 4 is cell mediated reactions. Now, the classification of immune reactions is based on the reactions. Like type 1 is IgE mediated immune reaction, that is an immediate reaction. And type 2 is again an antibody mediated reaction, but it is the antibody here is IgG and IgM. As we know, there are 5 types of Ig immunoglobulin, that is IgG, IgA, IgM, IgD, IgE. So here we have, in the case of type 1, we have IgE, and type 2, we have IgG and IgM, that is generally cytotoxic immune mechanism. Ig3 is antigen antibody complex. Due to the reaction of antigen antibody, formation of antigen antibody complexes, we call it as immune complexes. This hypersensitivity reaction occurs. And in the case of type 4, it is a T cell, that is cell mediated immune reaction. All the other 3 are antibody mediated or humoral mediated, whereas type 4 is cell mediated immune hypersensitivity reactions. So type 1 we also called as immediate type of hypersensitivity reactions. We also called as allergy. That is a normal terminology, as we all know. As a layman, we all know what is allergy. But here the antibody is IgE antibodies produces by plasma cells in response to stimulation of Th2 by antigens. We have Th means T helper cells. We have different types of T helper cells. There are 2 major type of T cells. That is thymocytes, that is T helper cell and T cytotoxic cell. We call it as short form Th and Tc. So among these T helper cells, we have different classes. Among that one class is Th2. So Th2, this plasma cells, IgE antibodies produced by plasma cell, which is in response to stimulation by T helper cells 2 by antigens. So this antigen that stimulate are called allergens. So antigens which stimulate Th2 cells are called allergens. So any type of allergy, we all heard about different types of allergy, like dust allergy, pollen, cosmetic, insects, some clothing, some drugs, etc. So anything can be act as an allergen to activate these T helper cells. So this exposure may be ingested, inhalation, injection, or direct contact. So it can be taken up one, can be inhaled one, either can be either injection or maybe some other direct contact. So it is, as it is an antibody related, hence this type 1 reaction is a systemic, that is not localized one. Localized, and difference between localized and systemic. Localized means, suppose a reaction which is taking place only in our hand. So itching is taking place, sometimes some insects or some set of type of worms, itching worms are there. So if it happily touches to that worm body or some other antigens which is related to that particular localized area, it will itching on the particular space, that is hands. It is not extrapolated to any other body part. If it is restricted to our own hand or our own local areas of the body part, it is called a localized reaction. Whereas some cases, suppose we are taking some jellyfish or a toxic and shellfishes, so it is entirely our body, the toxin is getting affected. So that is systemic. Suppose if entire body is getting affected, we call it systemic. And if only a portion of our body is getting, the reaction is occurring in a portion of our body, either hand or fingers or on leg or on face, we call this localized hypersensitive reactions. So, but in the case of type 1, it is a systemic. It will affect all over our system. So it is or can be localized to a specific target organs like rhinitis, asthma. It is only in the nose and lungs. The asthma and the rhinitis, it only affected to the respiratory system, not other part of the body. Whereas in the case of systemic case, it is called anaphylaxis reaction. And when the hypersensitive reaction is systemic, if it is affect to the entire body system, we call it as anaphylaxis reactions. So type 1 can be either systemic type or it is a localized type. So this is there should be in type 1 hypersensitivity, the lag is very short. That is the exposure to an antigen, whereas the exhibition of clinical symptom is taken within no time. So this is based on the mediators which is produced by the mast. With antigen, it will evoke an immune reaction by an mast cell which is present in our body. This mast cell produces, due to the evocation of some immune responses, the mast cell produces certain mediators. That mediators resulted in the production of an array of chemical reactions which occur in our body. That leads to hypersensitive reactions. So this we call it as an immediate type because the mast cells react immediately by producing chemical mediators. So that resulted in array of chemical reactions. That is called, hence it is called immediate type of hypersensitive reactions. So the type 1 hypersensitive reaction is further divided into anaphylaxis and atopy. Let's look on what is anaphylaxis. So anaphylaxis is an acute reaction. As I already said, it's a systemic reaction. It is affect entire our body. It is potentially fatal, and manifestation is systemic, and it is an immediate type of hypersensitivity reaction. So how does it occur? So here we can see in this figure the allergens which come and bind onto the antibody, that is allergen specific Ig antibody. Immediately when it comes and bind to the antibody, it will produce a signal inwards inside the mast cells to produce, resulted in the degranulation of mast cells. So this degranulation here we can see in the figure, we can see the some chemical media. After the degranulation, some chemical molecules are produced by the mast cells. So this degranulation result in the production of certain mediators or chemical compound that result in the anaphylaxis reactions. So as I said, there is an antigen comes and binds with the T helper cells. So T helper cells activate mast cells. So mast cells, so on exposure to the antigen on the IgE portion of the antibody, that will help in the that will come and cross-link between the, as we said in the previous slide, it will come and cross-link. Sorry. If you come, this allergen will come and cross-link between the IgE antibody. That will produce a signal, and with degranulation, that signal will help in the give an instruction to mast cells to produce degranulation of basophils and as well as the mast cells, and that resulted in the production of pharmacologically active mediators within few time. Within minutes, it will produce mast cells. It will produce chemical mediators. So this binding of this allergen to IgE we call it as sensitization. That is a priming dose that resulted in the degranulation and production of chemical pharmacological active mediators. So this the initiator or a binding of this antigen to IgE that resulted in the anaphylaxis reaction. That is a key role takes place in the initiation of anaphylaxis reaction. This initiator, we call it as allergen. Those who activate the response, we call it as allergens. So allergens can be either pollens, proteins. The major allergens are proteins like foreign antigens like serum, vaccines, egg, milk protein, nuts, drugs. Similarly, the venoms, which is due to the insects bites, and snake bite, that is wasp, bees, ants, etc. Some dust mites, some mold spores, and animal hairs, etc., dander, etc., are the major allergens which can evoke this type of anaphylaxis reactions. So we have seen it still. How does it occur? I mean, totally, what is the mechanism? How does the mechanism of hypersensitive reaction occurs? We are going to see. So this is the allergen, as we said. This is the IgE antibody which is present on the mast cells. So this Ig antibody, which is presented by the B cells, will bind with an allergen. So that allergen will bind in between the Ig antibody. So this Ig antibody in turn, along with the CD4 Th2 cells, Th2 cells have a CD4 marker over here. So generally the T cells have CD4 marker and the TC cells have CD8 markers. So this T helper cells always have CD4 markers in their surface. So along with an interleukin 4 chemical mediators, so there will be a total of reaction between B cells and T cells is occur that resulted in the formation of a memory cells and plasma cells. This plasma cells produce large amount of antibodies here, and this antibody are specific, that is they bind onto the sensitized mast cells, and when the allergen comes in again in contact with this mast cells, there will be degranulation occurs. Due to degranulation, there will be a large production of vasoactive amines that resulted in smooth muscle contraction and blood vessels. These vasoactive amines acted on smooth muscles, small blood vessels, mucous glands, blood platelets, sensory nerve endings, eosinophils, etc. And it will produce some chemical reactions or immunological reactions inside these cells. And also here we have memory cells. When a second time the same antigen comes in our contacting body, this reaction won't occur. Directly this antibody will come and bind to the muscle. The reaction is much more easier than the initial priming dose. The second dose will be, that's what it is quite fatal, because directly it is coming in formation of mast cell degranulation and production of vasoactive amines and certain chemical mediators. So these are the initiator, we call it as allergens in the case of anaphylaxis reaction. This is just sting B, that is the toxin which is stinging onto our body. This is the pollens or some other allergens. These are the initiation of initial anaphylaxis reactions. In skin, it is like poisonous plants, animal scratches, pollen, latex, etc. Injection, that is by means of bee stings, and ingestion by means of, that is which is taken inside our body by medication, nutshells and shellfishes. And again inhalations like dust, pollen, mold, mildew, animal danders, etc. can be leads to allergic reactions. So I said there are some chemical mediators which help in the anaphylaxis. So the chemical mediators are histamines, prostaglandins. There is an A is there. So histamines, prostaglandins, similarly serotins, thromboxane, leukotrienes. These are the major chemical mediators which is produced by histamines produced by mast cells and degranulation of basophils. Prostaglandins produced by neutrophils. Serotonin is again produced by mast cells and platelets, and thromboxane is produced by platelets, and leukotriene is produced by leukocytes. So these are the chemical mediators which help in the hypersensitivity 1 reactions. We learned about the anaphylaxis of type 1 hypersensitivity. Now we are moving on to atopy. It was coined by a scientist called Coco. So this is similar to hypersensitivity that spontaneously in human, but it is not fatal. As anaphylaxis reaction, atopy reaction is not fatal, but and also it is not systemic in nature. It is generally in local manifestation. As I said, itching, sometimes a plant, this poisonous plant, itching type of plant, which will come and touch to our body part. Suppose it is in our hand, or suppose in our face, that the itching or reaction is taking place only to the particular portion it will which comes in contact. Otherwise it won't affect the entire system or systemic, it won't attend the systemic reactions. So it is a recurrent reactions, and it is not fatal, and it's a local manifestation of immediate hypersensitivity reaction. So this is a local manifested reaction is atopy. So it is localized to specificity involved in epithelial surface of site of antigen entry. When majorly this type of reaction is occur on the skin or epithelial cells, that is it will limited to few minutes or hours will scratch. Sometimes it will, like suppose some mosquitoes bite, some wasp bite, you scratch it for some times, the swelling occur, for some times later it will be diminished, it will be vanished. So it is also mediated by Ig antibody. So it is this atopic individuals. Some individuals are very much atopic. We are prone to specific allergic reaction like dust and all. They are prone very frequently. It will come. So sometimes people, can exhibit allergy for months or days, but it won't affect her. It's not fatal. It won't lead to death and all. So this kind of reaction is against normal individual who the allergens sometimes there are few people are allergic to specific things, so which normally human beings are not. Among a few category of people are allergic to, that is sort of infection is atopic reactions like asthma, rhinitis, atopic dermatitis, etc., and urticaria. These are the common manifestation of atopy. So when compared to anaphylaxis, atopic reaction is not much fatal. So what are the how we can manage this anaphylaxis reaction? Sometimes antihistamines that block the H1 and H2 receptors on the target cells. Next is chromoleon sodium that blocks the influx into mast cells, calcium influx into mast cells. Theophylline that is prolongs the CAM level in the mast cells by inhibiting phosphodiesters, which cleaves the CAM to 5-adenosine monophosphate. And next is epinephrine, or we also call this adrenaline, that also have action on CAM production. And next is cortisol. So that reduces histamine level by blocking conversion of histidine to histamines by stimulation of mast cells production of CAM. So these are the major action on some medicines, which like antihistamines, corticosteroid, epinephrine, etc., to reduce the anaphylactic reactions. So here we can see certain tests which we are generally doing to find out whether the allergic reaction is there or not. So suppose we have dust allergy, sometimes we have mite allergy. We don't know what sort of allergy. Sometimes we get itching very frequently. And when we go to doctor say it could be due to the places where you sleep, it could be due to the place where you sit, it could be due to the environmental where you have some mite or dust, or some plants which is not suitable for you. We don't know what it is. Some trees, their pollens. So there are n number of allergens which can be around you, but we don't know which one is prompting you to have this kind of allergy. So in order to, we need to have an allergic test. The specific skin site either by intradermal injection or supra scratching. So this allergen can be injected in our body. We can wait. There are an array of allergens can be injected. We can wait for some times, and we can look for. In this picture, we can see a little bit reaction. We can see in the grass, which is marked as grass. So some grass is giving him some sort of reactions. Other places it is very nearly less. We can see this edema or swelling only in the grass portion, which is injected with the allergens. So that is individual allergic to substance subject injects local mast cell degranulate producing wheel and flare response within 30 minutes. So we call it as wheel and flare reactions. That is when within 30 minutes we will give an array of allergen reaction injection to body, and we look for what all the allergens we are reacted. So that is due to the local mast cell degranulation that reaction occurs. So within 30 minutes we get the result. And when it is similarly, if you can see here, it's some because swelling is there. We call this wheel flare reactions, and within 30 minutes we get the results. So other methods other than the skin test, we have radio immunoabsorbent assay test, and also we have radio allergoabsorbent tests, immunofluorescence method tests, ELISA, etc. can be done to test these allergic reactions. So what are the clinical manifestation of allergic reaction or anaphylaxis reaction? It's an acute life-threatening reaction. Hence, the symptoms depends on the level of hypersensitivity and amount of allergen which we have taken and the site of exposure. So these are the factors which is affecting the symptoms, like the amount of allergens, diffusibility, how fast it is getting in our body and diffusible in our body, and the site of exposure. Multiple organ systems are usually affected, including skin, respiratory tract, and cardiovascular system. Skin, it is pruritus, flushing, urticaria, and angioedema. In respiratory tract, it will be bronchospasm, laryngeal edema, etc. In cardiovascular system, it is hypertension and cardiac arrhythmias. So sometimes death occurs due to laryngeal edema and intractable bronchospasm and hypertensive shock, or cardiac arrhythmias is developing within 4 to 2 hours. So death is due to some laryngeal edema and some cardiac symptoms and bronchospasm, etc. Now we are moving on to the type 2 cytotoxic reactions. As we said, it is also an antibody mediated, but the antibody is different. First in reaction, it is IgE antibody. Here it is due to IgG and IgM antibody. So here the cytotoxic reaction is mediated by antibody directed against antigen on the cell membrane and activate complement, thereby causing antibody mediated destruction of cells. So in this reaction, this IgM and IgE antibody, which attack on the cell membrane surfaces protein, which activates an array of complement and resulted in the destruction of cells. Cell membrane damaged by membrane attack complexes during the activation of complement. This complement, it will activate a protein, array of protein called the complement. And the final product of complement is MAC, or membrane attack complexes, or membrane attack factors. So that will help in the action against cell membranes, and leakage of cell membranes occurs due to the action of complements, resulted in the degradation of or cytotoxic reaction or damaging of cells. So this reaction evokes by a combination of IgG molecule and IgM antibody molecule. They are still against the cell-fixed antigen. So orals, or the circulating antigens throughout our body. whatever it is. Maybe it is fixed on the certain cell surfaces or certain antigen which is circulating in our body. In both conditions, the cytotoxic reaction by IgG and IgM antibodies can occur. So here the major activation is the antigen antibody complex activate complement systems. This complement system produce a MAC, that is membrane attack complexes, resulted in the leakage and death of the cells. So this is a mechanism. This is a, suppose we have a cell surface antigen, target cell surface antigen, antibody which come and bind. Once the antibody binds on this antigen, it will activate the antigen immune complex will activate this antigen. This is antibody immune complex is formed. That in turn activate complement system, and this complement system finally produce a MAC complex. There are two ways a complement can act. We call it as alternative pathway and a classical pathway, and both pathways resulted in the formation of a membrane attack complexes that will attack the membrane by producing perforations on the surface of the membranes, and that resulted in the oozing out of the membrane, cell substances, resulted in the death of the cells. So, and finally, it either can lead to phagocytosis. Also, oxidation and phagocytosis can be finally resulted. So, either it is the membrane attacks complexes formed and osmotic lysis occurs, and also it can be destroy the antigen by means of opsonization and phagocytosis. So, in type 2 hypersensitive reactions generally occur in the blood grouping or blood transfusion reactions. It's very, very important because here the cell surface antigen on each red blood cells. In the case of we all know about the blood grouping. In a person who is having group A is having antigen group A and antibody B, and that is anti-B, and a group B persons will have anti-A and antigen B, and group AB is not having any sort of antibody, but they have antigen A and B, and group O is having antibody A and B, but they don't have any sort of antigens. This is a molecular or antigen based classification of blood grouping systems. So general, as you know, this is a cell surface antigens. This is A is a cell surface antigen. This is in the case of red blood cells, A group individual. The B group individual, they have B group of antigens. In the case of AB group, they have a mixture of A and B group. And O group, they don't have even antigen, but they have only the antibody against those antigens. So this is so hence, due to the presence of this A antigen, we cannot give B because here they have anti-A antibody. So hence we cannot give them the B blood cells. B group blood cells blood cannot be transferred to A group persons, and similarly vice versa, A group blood cannot be transferred to persons individual with a B group. But AB group is their universal acceptance. They can take blood from either of both. But O are universal donor, they cannot take any type of blood from both A and B group. So this is the conditions. So type 2 is act on cells causing damage to cells, and along with the action of complement resulted in hemolytic disease of transfusion reaction or hemolytic anemia. So this wrong blood transfusion. Suppose anybody is getting A group person is getting wrongly, they're receiving blood from a B group individual. So that result in blood transfusion reaction. Here, the reaction is this antigen-antibody complex is formed, the complement is acting. Immediately, it will targeted and kill the A group antigen, which is a transfusion, the antibody which is come from the other patient will kill the blood, already blood cells is with antigen antibody. Suppose we are taking antigen from the blood from the B group individual. The B group individual blood system can damage to the A group, the receiver's blood systems. So that is called a transfusion reaction or hemolytic anemia. Antibody-dependent cell-mediated cytotoxicity, that is ADCC, is a mechanism which involves the binding of cytotoxic cells with the FC receptors in the FC binding part of the antibody coating onto the target cells. This antibody coating the target cells also cause a destruction by acting on opsonin. There are two ways it can be directly. It can kill the cells by MAC complex, or else it can lead to it direct and the cells to engulf or opsonin by the targeted cells by certain cells, macrophages like cells, and kill the target cells. So, as I said before in the previous slides, I said like how a blood transfusion reaction is if a wrong blood transfusion reaction is deleterious to an individual. So, antibodies here, a large number of proteins like glycoproteins are on the surface of RBC, like A, B, and O group antigens are particular importance. Antibodies to these antigens are called isohemagglutins or IgM class of antibodies. So when transmutation is mismatched occurs, a transfusion reaction takes place due to destruction of donor RBCs through isoagglutinin against the foreign antigen. As I said, the donor RBC is getting hemagglutinized against the foreign antigen. The clinical manifestation resulted in the hemolysis of the donor cell by antibody and problem. The donor cell is getting hemolyzed. If this reaction, if suppose wrong transfusion resulted in the hemolysis of the donor cells. Next is another cytotoxic reaction is erythroblastosis fetalis conditions. As you all know, there are two types of antibodies, Rh plus and Rh minus antibodies. Suppose a person, a lady marries, Rh negative person married to a Rh positive individual. Their first baby, if it is Rh positive and if the mother is Rh negative, the first conceive is not a problem because there won't be any mixing of blood between the mother and baby, because it is entire through the placenta carrier, the food is transferring. There is no blood contact between the mother's blood as well as the baby's blood. But when the delivery occurs, naturally there will be a mixing up of blood between the parent and the mother and baby. Soon after the delivery, because the baby is Rh positive and the mother is Rh negative, in that condition, Rh positive baby will give its antigens to the mother, so that the mother will develop a Rh positive antibody. So during the delivery time, that's the first delivery time. Suppose the lady is again getting pregnant or with an Rh positive fetus, immediately that fetus is getting eliminated due to the already existing Rh positive antibody, which is formed during the first delivery. So this condition is called the erythroblastosis fetalis condition. This leading to hyperbilirubinemia and anemia, even just death to the baby. So this is a condition. The time of this is Rh antigen. The baby is with Rh positive. The mother is Rh negative. So it is in the maternal circulation. It is no way it is during the pregnancy time. It is no way it is getting conducted with the mother. But during the delivery time, this RH first pregnancy, the first pregnant delivery, this antibody is getting, antigen is getting mixed with the mother's blood. So there will be an RH specific antibody will be produced during that time. So this plasma cells will produce antibody as well as they will have a RH positive memory cells also will be there. That is the same during the second pregnancy. The RH positive is baby is again she's carrying an RH positive baby. The memory cells immediately will recognize this RH positive antibody, and that will resulted in the elimination of this baby from the womb. So this resulted in the development of erythroblastosis condition. This is a cytotoxic mediated or type 2 hypersensitivity reaction. So type 2 hypersensitivity reaction is called a cytotoxic reaction. This involves IgM or IgG antibody mediated reactions, or this transfusion reaction, and this hemolytic disease of newborn is an examples of hypersensitivity reaction. In addition to that, drugs can induce hemolysis. That is, drugs such as penicillin, quinidine, phenylephrine, they can induce hemolysis of red blood cells. So these drugs can attach onto the, we all know that is a membrane surface antigen is getting killed or getting eliminated during the cytotoxic reaction. Here drugs is attaching on the cell surface of the red blood cells, and that stimulate the formation of IgG antibodies that resulted in the hemolysis. And quinacrines attached to surface of platelets and induce autoantibodies that result in the lysis of platelets causing thrombocytopenia. Rheumatic fever is another example of type 2 hypersensitivity reactions. In this condition, antibodies are produced against group A streptococcal that cross-react with the cardiac tissues and activate complement. And we know the release of complement is to MAC and damage of cardiac tissue that resulted in the cardiac tissue. That is also another example of cytotoxic 2 reactions. Now we are moving on to type 3 hypersensitivity reaction, which is called immune complex mediated hypersensitivity reaction. So this immune type 2 reaction mediated by antigen antibody immune complexes induces inflammatory reaction in tissues. So type 3 reaction causes inflammatory reactions in tissues. So it is called a immune complex hypersensitivity reaction. In many situations, reaction between various antigens and antibodies in the body give rise to formation of immune complexes. So we have several instances, and antigen antibody complexes can be formed in our tissues or in our body. The normal cause, these immune complexes are normally removed or by mononuclear phagocytic systems through participation of RBCs. Generally, this immune complexes getting finally removed by RBCs. So this type 3 hypersensitivity is known as immune complex reactions. 3 to 10 hours after the exposure to antigen this reaction time is around 3 to 10 hours once after onset of exposure to antigen. So example is ARTHUS reaction and serum sicknesses are the type 3 hypersensitivity reaction. So here the individual organs including other organs are getting infected. Antigen causing immune complexes are exogenous and endogenous. There are two types of antigen which can cause immune complex reaction. One is exogenous antigen, other one is endogenous antigens. Here the antigen combines with antibody within the circulation and forms immune complexes. Here antigen is combines with the antibody within the circulation and form a immune complex formation. So wherever the body, this immune complexes get deposited in various parts of our body. Once they get deposited various part of the body, that as we know, antigen antibody complexes can activate the complement system. So these polymorphonuclear cells are attracted to, during this activation of complement resulted in the attraction of polymorphonuclear cells to that site, that resulted in inflammation and tissue injury. So once this antigen antibody reaction is formed, they form an immune complex. They get deposited in certain part of the body, which finally attract complement system. This complement system in turn attract polymorphonuclear cells towards that site, which resulted in the inflammation and the tissue injury. So this is a mechanism. The antigen, here we have B cells, it produce antibody. Antigen antibody complexes is formed, that is called immune complex is formed. This immune complex, once it is formed, it attract complements. Once complement is we know MAC system will form, directly it can cause tissue damage, or else it activate the neutrophils, or else polymorphonuclear cells. And finally, when the neutrophils comes, we know there will be phagocytosis and opsonization can take place, resulted in the tissue damage. So examples are ARTHUS reaction and serum sicknesses are the two typical manifestation of type 3 hypersensitivity reactions. So here generally what happens is, we all know the antigen antibody complex is formed, they fix a complement. So this activation of this complement forms there are different array. I said there are two pathways in which the complement is getting activated, classical pathway as well as the alternate pathway. So both ways there will be some reactive molecules will be formed due to the cleavage of certain complement systems. There are two major cleavage systems is formed, that is CA3 and CA5 cleavage compound. They that is the C5 component and the C3 component get cleaved into C3A and C3B. In the C3A is getting into the system and stimulate the production of vascoactive amines. Similarly, C5A is also resulted in the stimulation of vascoactive amines. And this C5A also help in the attraction of neutrophils to that site, that resulted in the phagocytosis reaction and tissue damage, lysis of tissues and damage of host tissue. So in addition to that, the neutrophil produces certain enzymes like neutral proteins, collagenase. There are other proteolytic enzymes like kinin forming enzymes, polycationic proteins, the reactive oxygen and nitrogen intermediate. This all resulted in the damage of local tissues and enhanced the inflammatory response. Not only the complement activation and the production of vascoactive amines, there are additionally proteolytic enzymes which is produced by various reactions are also help in the damage of local tissues and inflammatory responses. Platelets aggregated by intravascular complexes provide another source of vascoactive amines and resulted in formation of microthrombi, which can lead to local ischemia. So this is also another way of action of type 3 hypersensitive reaction, that is platelet aggregation, and finally it can formation of microthrombi, which can lead to local ischemia reaction. So one of the major manifestation of type 3 reaction is ARTHUS reactions. So this is the tissue damage by the deposition of antigen anti-complexes resulted in the production of vascular occlusion and necrosis. So there will be a vascular occlusion and necrosis is a major active reaction which take place in the ARTHUS reactions. So hypersensitivity pneumonitis is a clinical manifestation of this ARTHUS reaction. So farmer's lung, cheese washer's lung, woodworker's lung, and wheat miller's lung are the examples of hypersensitivity pneumonitis associated with the different occupations. So this is an occupational based hypersensitivity reaction. All resulted in the occlusion in the that is vascular occlusion in the respiratory tract. We call it as in a different farmer's lung. It is associated with the occupation, and cheese washer's lung, that is cheese. It is related to cheese manufacture, and woodworkers is related to again to other occupation, woodworkers, and wheat miller lung. So again another type of occupation that is associated with the wheat and miller production. So here in this all this condition, there will be a chance of inhalation of fungus or bacteria present in different product that is handled by the infected people. That resulted in the occlusion of the respiratory tract. So another reaction, which is example of type 3 infectious serum sickness, is a systemic inflammatory reaction caused by the deposition of immune complex in our body. This condition is manifest after a single injection of high concentration of foreign serum. It appears to be in within 2 to 3 days, like injection of foreign serum or certain drugs like penicillin. So the single injection acts as a both priming and shocking dose. Here in the case of hypersensitivity 1 reaction, they require a first initial dose, initial priming. Then it is second, in the case of immediate hypersensitivity reaction, though it is an immediate hypersensitivity reaction, there should be a two dose are required. Initial dose and priming dose, and second dose is required. That is here in this priming dose is where shocking dose is required. Whereas in this case, one priming dose itself can cause deleterious reactions. The symptoms are fever, lymphadenopathy, rash, arthritis, splenomegaly, eosinophilia, etc. Disease is self-limited to clears without sequelae. So after a few days, it can be cleared out without any circulation. Serum sickness considers an immediate hypersensitive reaction because the symptoms appear immediately after the formation of immune complex. So it can, though it is a typical delayed, immediate hypersensitive reaction due to IgE antibody, but it is a immune complex immediate hypersensitivity reaction, we can say, because it's taking place within 2 to 2 weeks. Immediately it can happen, but it is not like a type 1 hypersensitivity reaction, because here there is only IgE antibody. Here it is not so. So hence the serum sickness can be considered, though it is not an immediate hypersensitivity, can be considered as an immediate hypersensitivity reactions. So another infections are that is systemic lupus erythromatous condition is another immune complex reaction. Rheumatoid arthritis is another immune complex disease, and good pasture syndrome is also considered as an immune complex disease. So formation of circulating immune complex contribute the pathogenesis of number of conditions other than serum sicknesses. So this sometimes the circulating immune complexes can lead to certain diseases. These are the examples of the circulating immune complexes, like some autoimmune diseases like systemic erythromatous condition, rheumatoid arthritis condition, good passes symptoms, all are a type of immune complex reaction. Drug reactions, allergic to penicillin and sulfonamides is also example of a drug-based immune complex disease reaction. Due to infections, also we can result in immune complex reaction like meningitis, hepatitis, malaria, trypanosomiasis, post-streptococcal glomerulonephritis is also an immune complex reaction, which is led by the infectious diseases. So what are the major diseases in type 3 hypersensitivity. We are just going through the nutshell of that. Hypersensitivity pneumonitis, glomerulonephritis, rheumatoid arthritis, and systemic lupus erythematosus or SLE are the major type of immune complex diseases. So, in again, this type 2, as we said before, atopic and hypersensitivity, anaphylaxis reaction. Here also we have, in type 3 also we have two major type of hypersensitivity reactions, local reaction and systemic reactions. This ARTHUS reaction, which is named after Dr. ARTHUS, is an inflammatory reaction. It is a local reaction in the localized site. It is a clinical manifestation of hypersensitivity pneumonitis. Again, the serum sicknesses is a systemic inflammatory reactions that is 2 weeks after injection of foreign antigen like urticaria, Allergy, eosinophilia, splenomegaly, and lymphadenopathy. These are the hypersensitivity reactions. So, hypersensitivity pneumonitis is an inhalation of antigen into lungs stimulate antibody production. So, subsequent inhalation of the same antigen resulted in the formation of immune complex and activate the complement. Glomerulonephritis is an immune complex reaction in the blood and deposited in the glomeruli and resulted in the damage of glomerular cells and resulted in the kidney failure. Ultimately, death can be occurred. Rheumatoid arthritis and other symptoms resulted in the deposition of this immune complex on the joints, resulted in the release of inflammatory chemicals that the joints begins to break down and becomes distorted. The joints, due to the inflammatory chemical, the joint begins to break down and become distorted. What is the reason which triggers is not well understood. Still, we can treat it with the anti-inflammatory drugs. Next is SLE or systemic lupus erythematosus, because generally why I have written SLE, because some of the major exams they ask for what is only SLE. They won't ask the full form, that is systemic lupus erythematosus. So the short form needs to be studied. So it is an autoantibody against the DNA result in the immune complex formation. So again, the trigger of this formation is still unknown. Many other though it is an autoantibody reaction, it is also comes under the hypersensitive reaction because it is formation of antigen antibody complexes and autoantibodies. Due to the autoantibody production, there will be antigen antibody complex formation is also occur. Here, the antibodies are formed against red blood cells, platelets, lymphocytes, and muscle cells. And here we can result in the formation of inflammation. Inflammation can result, but still it can be reduced due to the glucocorticosteroid drugs and immunosuppressive drugs. This is a typical SLE symptoms. Now, finally, we are moving on to the last hypersensitive reaction, that is cell-mediated type, that is T cell-mediated type, that is type 4 hypersensitive reaction. Here we call it as delayed hypersensitivity reaction. It starts hours or days after primary contact. After the priming dose, it will take days to react with the second dose. So the reaction is characterized by a large influx of non-specific inflammatory cells, in particular macrophages. It is mediated through cell-mediated immunity. The reaction occurs due to the activation of specifically sensitive T lymphocytes rather than antibodies. So here the T lymphocytes activation is very mandatory than antibodies. It was initially described by Robert Koch in the formation of tuberculin reaction against tuberculosis. So this is later it is found out that it is a various pathological condition is due to delayed reaction, hence it is named as delayed hypersensitivity reactions. So mechanism of delayed hypersensitivity or type 4 reaction is, and DT's response begins with an initial sensitization phase of 1 to 2 weeks. So Initial sensitization itself is taken 1 to 2 weeks after the primary contact with an antigen. Here, unlike immediate hypersensitivity, TH2 is reactive. Here it is TH1 subtype is reacting in the case of delayed hypersensitive reactions. So T helper cells type 1 is in the action of DTH, whereas in the case of type 1 reaction, it is TH2. So TH1 subtypes CD4 are the cells activated during the sensitization phase. So the first sensitization phase, the TH1 is getting activated. In fact, TH1 CD4 get markers is getting activated. A variety of antigen presenting cells, APCs. There are different antigen presenting cells in our body, like macrophages, dendritic cells, etc. So similar, this antigen presenting cells and especially in the antigen presenting cells, when it comes to the respiratory tract, we call it as Langerhans cells. So the Langerhans cells and macrophages are getting activated and resulted in the formation of DTH response. So, Langerhans cells are present on the dendritic cell. It is present in the lung cell, respiratory cells. Langerhans cells present on the skin cells. So, Langerhans cells, which is present on the skin cells, that is the antigen presenting cells, they pick up antigens and enters into the skin and transport into the regional lymph node, and where T cells are activated by antigen. So, this antigen presenting cells picks up this antigen and present it to the T cells, which is present, which is in the lymph node, and activated the T cells by this antigen. So delayed hypersensitivity is a function of T lymphocyte antibody. It starts within hours or days after contact with an antigen, or often lasts for days. It can transferred by immunologically committed T cells, not by serum. It is not transferred by serum, but it is by immunologically committed T cells. The immunological response of such reaction is by various type of microbes like Mycobacterium tuberculosis, viruses, fungi, parasite, etc. can evoke type 4 hypersensitivity reactions. It is activated by T lymphocyte, release cytokines and macrophages activation, and T mediated cytotoxic reactions. So the activated T lymphocyte acted in two ways, like release of cytokines and macrophage activations. So once cytokines is released, that cytokines will activate the macrophages, and that resulted in certain immunological responses. Then T cell mediated directly T cells can kill the infected pathogens. So suppose it's a viral infected cells. So directly T cells can damage that viral cytotoxic. It becomes cytotoxic to particular infected cells. So one of the example is type 4 hypersensitivity reactions, tuberculin response. An injection of tuberculin beneath the skin causes reaction in the individual exposed to tuberculosis or tuberculosis vaccines. So this type of tuberculin reaction is help to diagnose the antigens against tuberculosis. So there won't be no reaction for those persons are not infected with tuberculosis or vaccinated, but there will be symptoms to that is skin reaction can occur in the case of persons which are infected with tuberculosis or not vaccinated with tuberculosis. So tuberculin reaction is helped to determine whether the persons are comes in contact with the tuberculosis pathogen, or they are taken the tuberculosis vaccine. So this is a positive tuberculosis test. So once the tuberculin antigen is injected to our body, so after a few hours we can see a bulging of this reaction. If it is this type of reaction occur, we can confirm that the tuberculin test positive, and patient is or either is taken up the vaccine recently or infected with the pathogen. Another infection is cell mediated, that is allergic contact dermatitis. And this is a cell-mediated immune response resulting in intensity of irritating skin rashes triggered by chemically modified skin protein that the body regards as foreign. Acellular fluid-filled blisters develop in severe cases. There will be fluid-filled blisters will be developed in severe cases, can be treated with the glucocorticosteroid. So this allergic dermatitis can be is a irritation and skin rash reaction due to cell-mediated immune response, but it can be treated with the corticosteroid drugs. This is an example of this contact dermatitis reaction. So next is graft rejection. That is once this is an again a cell-mediated hypersensitivity reactions. We all know, like recently, very much medical advance is taking place due to the transplantation therapies. So transplantation means taking up organs from some other individuals or our own body part replaced with our own body parts, or it can be taken from some other unrelated species. So many type of tissue grafts or grafting reaction is going on. So the graft acceptance or rejection is based on our immune reactions or immune type, which is how our system react with this graft. So rejection is another type of the cell-mediated hypersensitivity reaction, that is rejection of the transplanted organs. The graft perceived as foreign and recipient undergo rejection. So this graft, when it is taken from another individual, our body considers it as a foreign substance and immediately getting rejected. So generally it is acted against the major histocompatibility complex on the graft cells, and the major histocompatibility complexes of the graft cell is considered as foreign, and immediately our body reject such graft. Rejection depends on the degree of the graft is foreign on the or to the recipient. How the recipient compatible with that graft, that is the yardstick of this rejection. So if our body is considered totally foreign and it's immediately it get rejected, and otherwise it stay for some days, later time it get rejected. So based on the type of graft we receive, the rejection occurs. So major type of DTH are contact hypersensitivity reaction and tuberculin type hypersensitivity reaction. Contact hypersensitivity reaction is a manifestation of delayed hypersensitivity occurring after sensitization with a certain substances. This contact, it is once it getting contact with a certain substance only we are getting under sensitization, and later it can result in delayed hypersensitivity reaction. This includes sulfonamide, neomycin, plant products, poison ivy, and poison of chemicals such as formaldehyde, nickel, and cosmetic soaps and other substances can leads to this hypersensitivity reaction. So this reaction manifests when a substance acting as a haptens enter the skin and combines with the body protein to become complete antigen, which person becomes sensitized. Here, in which the substances acting as haptens or allergens enters the skin and combine with the body proteins and become complete antigens to become person get, because here the complete we know antigens are require certain criteria for to become antigen. So if the antigen is not complete, we call it as haptens. So a hapten cannot be act as a complete antigen. So what happens is when an haptens that is, we call it as semi-antigens or partial antigens, when enter into our body, it combines with our body proteins and become complete antigen, and later the person get sensitized. So here the contact dermatitis on the second exposure to the same antigen resulted in the attack of cytotoxic T cells and cause damage to the skins, like itching, erythema, eczema, necrosis, etc. are the symptoms of this contact dermatitis. Next is DTS, tubercle type of hypersensitivity reactions. This tubercle reaction typically example of delayed hypersensitivity reaction against microorganism, which is being used as diagnosis of this disease. As I said, the tubercle test is carried out to determine whether the individual has been exposed to tuberculosis or not. Here in this test, we are taking a small amount of tuberculin protein, and which is tuberculin protein is present on the cell wall of Mycobacterium tuberculosis, and it is getting injected intradermally. We are waiting for 40 to 78 hours to detect it positive or not. So I showed this reaction. I just explained it is once again. That's all. So this is the tuberculin test. If it is positive, we are getting this allergic reaction like this. So some patients will get negative even though they have positive, because suppose they have taken already with the corticosteroid drug, it will get negatives. So as I said, graft. So just for your information, I showed this graph. What is an isograph, what is an allograft and xenograft. Isograft is a graft from the same individual, genetically identical siblings or clones. Allograft, it is from genetically different species of the same member, and xenograft, it is entirely different species. It is called a xenograft. So this is a type 4 hypersensitivity reactions. First is sensitization phase. That is when bacteria comes in contact, it will presented by the dendritic cell, antigen presenting cells. Like I said, it is Langerhans cells in this case of skin. So antigen presenting cells. It will find this bacteria, and it is presented to Th1 cells, and Th1 cells will produce large amount of chemical mediators, that is, and which will again resulted in the formation of few more T helper cells, and a large amount of chemical mediators again will be produced. So this is a sensitization phase. There are two in every pathogenesis reaction, in every immune reaction, there will be two phases. That's the first is the initial phase, then this is the effector phase. That is, effector means acting phase. Initial phases, that is sensitization phase, is the production, the formation phase, how the immune system is getting formed. Then the action phase, that is the effector phase. The action is the effector phase. Here the T helper cells, that is along with the tumor TNF alpha and interferon gamma, which will act on the resting macrophages. It activate the macrophages, and it act on the MHC, and finally the activated macrophages will find out along with the class 2 MHC molecule, TNF receptors and other things. It's getting activated, and production of a large amount of free radicals, oxygen radicals, nitric oxide, etc. So in effector phase, it produces, in the case of Th1, it produces alpha, beta, TNF, and interferon gamma, TNF alpha, interleukin, L3, and IL-8, macrophage activator factor, and microphage initiation factors, etc. It will be produced, and it will initiate macrophages also. Finally, once the macrophage is getting activated, it will result in the engulfing of particular bacteria and killing that bacteria. So macrophages activation resulted in the formation of MHC class 2, TNF receptors, oxygen radical, and nitric oxides. Those also help in the degradation of this antigen, which is encountered, finally get into the macrophages. So this resulted in the end of this session. Before going to the winding up the sessions, we are just have a look on what are the type of reactions. This 4 type of reactions. Here, if it is say, once an allergen comes in contact, there will be a degranulation of the mast cells and produce phosphoactive amines. So generally, it is mediated by IgE, hypersensitivity reactions. In second case, antigen-dependent cytotoxic reactions. Here antigen is coming and binding onto the Fc receptor for cytotoxic T cells, which is presented with the, that is antigen binding cells. So the target cells will be antigen antibody complex will be formed on this target cells, and later it will produce, this is called surface antigen. So this surface antigen is attached with the antibody and form immune complex, and this immune complex will activate the complement, and the complement mediated activation resulted in the cytotoxic hypersensitivity reactions. And here also we have immune complex reaction, that is this is a typical immune complex reaction, Ig and Ig antibody, that is resulted in the complement formation. Complement formation resulted in the formation of MAC, this membrane attack complexes, or else activation of neutrophil resulted in the apoptosis or engulfment that resulted in the hypersensitivity reaction. Next, finally we have T cell mediated immunity, where the antigen is taken up and presented by the antigen presenting cells, and later by to initiate the T helper cells 1, and that activate again the macrophages, and the macrophages resulted in the formation of certain chemical mediators as well as nitrous oxide radicals, and resulted in the killing of pathogens or engulfment of pathogens by cytotoxic reactions. So here these are the major, that is, this is the antigen Ig mediated, this is the antigen antibody mediated IgG and IgM mediated reaction. This is a typical immune antigen antibody complex mediated reaction, and this is a sensitized T cell mediated reactions. So this is the conclusion of hypersensitivity reactions. Thank you.